To Mike. Thank you very much, Mike.
Yeah.
Amazing chair, as usual.
Cheers,
and
thank you.
jumping and stepping in at last minute. Thank you, panel. And now it's time to move on to our next panel, which, as you notice, there's a theme around Genesis. It's highlighting all the hot topics that happened in the year. And I think one, in the vein of this very hot topic, Genomics is clearly holding a good step. So I'd like to invite our next panel, chaired by our fantastic Adrian Ibrahim, Head of Technology Transfer and Business Development at the Wellcome Genome. Wellcome Sanger Institute. I'm getting a bit tired, that's the end of the day. Anyway, and so to address towards the genomics-driven healthcare system. So obviously, genomics, huge topic and of interest for the UK, for the technology excellence and the capacity of translating science and generating innovation. But remains still lots of questions on this adoption, and so we have a really good, diverse panel to discuss that in the next 50 minutes. So I think I'll take the line as with Mike, so if you have any question, we'll keep them for probably the end of it, so think of them, and about 10 minutes before close, I'll stand and you can raise your hand. Is that okay?
That's brilliant, thank you.
Adrian, over to you.
Thank you very much. Before we start, just a big thank you for coming and for staying. You know, this is the first time since the first lockdown that I've taken the hour's journey from Cambridge into central London. And as great as Zoom is, I got zoomed out about 15 months ago. You know, and there's no denying you lose quite a quality from not having face-to-face, so thank you. That's appreciated. We're going to spend the next 50 minutes talking about the impact that genomics, I think genomics quite broadly, genomics, biodata, informatics, machine learning, is having in healthcare, both at the patient interface, but also in the R&D setting. You know, it's always a better time for genomics to contribute than yesterday, and it always will be. So we have a fantastic panel. We have an incredibly, in fact, this is an unusual level of diversity for any conversation that has the word genomics in. And we have representation from big pharma, from SMEs, from healthcare data science, and from investment at different stages of company maturity. I did a quick kind of mental back of the envelope thing, and I reckon there's probably 100 years of genomics expertise on this stage. I wasn't going to say about the distribution, but please do bring questions and exploit their presence. I'm not going to introduce them. I'm going to ask them to introduce themselves each time they start and have a first question. But I'm going to start with our investors on this side of the stage. There are ever more companies or prospective companies out there. You see an enormous number of plans and propositions. And I guess my first question is, you know, what are you seeing in terms of genomics technologies, genomics platforms, genomics-orientated business plans and models that excites you and thinks, you know, this can impact a market, this can maybe create a market, this can disrupt a market? And if I start with you, Anya, and just also ask you to introduce yourself as you start.
Sure. Can everyone hear me? All right. Okay. Hi, everyone. I'm Anya. I'm the head of Illumina Accelerator, which is effectively the investment arm doing pre-seed, seed stage investments on behalf of Illumina. And we have two goals with this. One of them is to really support early stage genomics startups globally, but also build out the genomics ecosystem. So that's effectively my role. But to answer sort of like your question in terms of what I'm seeing sitting at Illumina is, you know, what's really interesting is single cell sequencing is exploding. So that's super interesting to see the resolution that might come through that in terms of, you know, looking at somatic clonal structures and how that can help with cancer therapeutics in the future, and looking at the ability to differentiate or looking at the heterogeneity of cells in particular. But the second aspect that I'm also seeing in my role is people exploring the dark genome, seeing that we have around 20,000 protein-coded genes that have been well explored, which consists of around 1 to 2% of at the human genome, what's happening with the rest? I think this is something that's we're seeing startups really explore, and we're excited to be backing some of them. And of course, you know, when you look at gene therapy, like one of the key concerns that has arisen is sort of looking at the safety of gene therapy. So we've backed companies who are looking at double-stranded DNA breaks that help to look at unintended off-target effects of gene therapies early in the process, so safety by design. So these are some of the aspects that we're seeing, we're excited about backing, but I know this is sort of like very much focused on human health, but I'm excited about what synthetic biology is, the revolution in synthetic biology and for genetics to play such a huge role in that in the future. But I'm going to pause now and allow Mike to speak.
Great, thanks. I'm Mike Anstey, partner at Cambridge Innovation Capital, which is a venture capital fund focused on IP-rich innovation coming out of the Cambridge ecosystem. Cambridge is a great place for investing in and growing genomics companies, and I think it's not a surprise that there's a number of people on this panel that actually have a presence in Cambridge. And so what we're seeing, I think, is world leading when we look at genomics as a space coming out of the Cambridge ecosystem. Between us, we'll probably cover all aspects of life sciences, because it's true that genomics is now embedded in every aspect of life sciences, and without genomics, I think there's a weakness in approaches. So a couple of areas that we're excited about. I think long-read sequencing will change things, and I'm really keen to hear this panel's view on how that's going to enable better genomic medicine and better support for patients. But I think that is game-changing, and great that Illumina has brought us to where we are right now. But with Nanopore and with PacBio and others, exciting things will happen, and I'll be keen to hear this panel's view on that. So that's one area that's important. I'm glad you mentioned the non-coding region, so that's also a really hot area. We just did an investment in a company that is looking at neoantigens arising from the non-coding area. And there's this really interesting finding that actually the mutanome may not be the primary source of immunological differentiation between a healthy cell and a cancer cell. And actually the non-coding region could be really where the right and best targets are. And that's paradigm shifting, and that turns the way we think about drug discovery on its head. So watch that space. I'm fascinated by the microbiome and TBD where that will yield, but I'm personally confident that will yield exciting results. As we move beyond FMT into defined bacterial therapy, you need deep genetic sequencing of the microbiome, and that will yield a whole bunch of exciting diagnostic applications as well as therapeutic applications. And then last but not least, liquid biopsy. Liquid biopsy is here, it's now. And whether it's early detection, whether it's looking at minimal residual disease assessment, whether it's long-term monitoring, ctDNA has opened up a world of potential, and hopefully will lead to better patient outcomes.
No, thanks very much, Mike and Anya. I have several questions. I'm just going to hold it and open the same question up to anyone else from the panel that wants to contribute in terms of the early technology you're seeing that really can have a paradigm shift in a healthcare setting.
I'll take a slightly different approach. So I'm Joanne Hackett. I'm the head of Genomic and Precision Medicine at IQVIA. And I think one of the things that's really quite exciting for me is being able to see that there's much more data being generated now. And because we have that data, and there's actually a lot of clinical data that's been in use for many years, actually being able to marry the two of those together and make diagnostics better and faster, cheaper, and earlier is much more exciting. So it's not necessarily a new innovation per se, but it's the ability for people to think slightly differently about that linkage aspect and having new technologies and using NLP in a slightly different way. So it's not genomics per se, but it's actually using the data that currently exists to get more and better out of genetics as a whole.
David.
I'll just add to that. So I'm David Atkins. I'm the Chief Executive at Congenica, which is a Cambridge-based genomic data company. We benefit from investment from CIC, and Adrian, who referred to me as an old person earlier, is our landlord at the Wellcome Foundation. When you think about technology, and for those of you who've been in genomics for 20, 30 years plus, if you go back to the late 70s, early 80s, really the transformational events were restriction enzymes, DNA sequencing, PCR. And and the move from the 70s to the 80s to the 90s, and the growth of genomics in the form of, say, infectious disease diagnostics, NIPT, well, actually not NIPT, but that whole $20 billion molecular diagnostic industry that quite often isn't thought of as genomics, but is very much a function of the sort of the methods and techniques that were developed in the 80s and early 90s. I very much agree with Joanne that the next phase, I think, is going to be less about tools and tricks. It's going to be about data. And it's going to be, in my opinion, about the diversity of data, but also the linkage of different data types to provide different insights. So I think the next 10 years, it's not going to be about who's got the best instrument, the best enzyme, best widget, the best tool. It's going to be who uses the data that becomes available and who generates the missing pieces in that data puzzle.
Thank you, David. I'm just going to come back to Anya and Mike. So, Anya, you talked about the power of single cell in the context of cancer. How do you see it adding value outside of oncology?
I mean, there are several sort of different applications, especially like when you look at Alzheimer's. There's a lot of research being done in the Alzheimer's space, even cardiovascular disease. So cell heterogeneity and having that resolution is really key to unlocking some of the aspects that we still don't know about and don't understand well. So there are several different, I mean, cancer is probably the one that's been studied the most, but there are several different indications where this is very applicable.
Thank you. Mike, on the dark genome, can you comment from what you've seen on challenges around the tractability of the types of targets that you'll see coming out of analysing the non-coding genome?
Yeah, so that's a big challenge. That's probably why we're not seeing that many therapeutics emerging yet. My view is that when you're looking at proteins that emerge in the cell surface, and those proteins that are a result of the non-coding genome starting to code, those can be great targets. So that's where I would start. Where I'd like this to go is I'd like this to go into cancer vaccines. And what's the leap between what we just described? Well, what we have evidence of is that some of the neoantigens arising from the non-coding region seem to be highly shared across individuals. And so I'm sort of trying to move away from highly personalized medicine, much more into off-the-shelf medicine. And that this source of neoantigens from the non-coding region could enable that. It's a big step, and you know it's quite bold thinking by some of the researchers, but I think that could unlock a whole new approach and make these drugs much more attractive.
Thanks, Mike. We're going to move on a bit now and bring Matt as well as Joe and David into it. And I think it'd be great if you could kind of speak to the audience about how your organisations are applying genomics in the clinical setting, decision support, but also in the R&D setting, and maybe more importantly even, what you see as the major barriers, the hurdles to adoption of genomic technologies, genomic services, genomics products. And Matt, maybe I'll ask you to kick off on that one.
Yeah, sure. So my name is Matt Beard. I'm a member of Ipsen, so we're a mid-sized pharmaceutical company. And my role is in licensing and partnerships. So I assess programs for strategic fit and scientific feasibility. And I can absolutely confirm that genomics and genomic approaches are key now to every modality. So everything from small molecules right through to the modalities where you'd expect genomics to be involved, such as siRNAs, antisense type modalities. We always look at genomic approaches to assess everything from target validity to patient segmentation. So for instance, if you're going for an antisense that's targeting a non-coding region, a regulatory region, perhaps giving you an exome skip or something like that, we really rely on a genomic analysis to look at epidemiology, what patient segment is going to benefit from that particular drug. So it really is a key part of our scientific evaluation. And really the biggest barriers that we run into at the moment for making more use of genomic approaches is availability the data and really interpretability of the data to non-expert people. So, you know, I'm a biochemist, cell biologist. I don't have a background in genomics. So at the moment, I really rely on review articles, reports, so global data, you know, those sort of synthesis of reports to get the genomic insights that I need to evaluate opportunities. And really, because I'm not an expert, I go to the classical ways to evaluate the strength of data. You know, I look for are there multiple sources giving me the same information? What sources do they source? Are they all citing the same publication, or is there a spread of publications? So really, the more accessible the information can be and the more verifiable it can be by non-experts, the more valuable it'll add to our assessments.
Thank you, Matt. I'll come back to you on some questions afterwards, but let's hear about the work that IQVIA is doing.
Sure. So IQVIA is the human data science company, and we've got trillions of clinical data points, which is very exciting. We have a huge amount of expertise in clinical trials as well, and an early investment in genomics from many years ago. And that was really one of the things that I think has pivoted the interest in genomics for IQVIA. And it's not, as I said previously, it's not necessarily about us developing new diagnostics and whizzy apps and things like that. We look to our partners for that. It's much more about being the plumbers. So I often say I want to grow up to be a ballerina, but a bit disappointing to become a plumber. But nonetheless, everyone needs a plumber in their life. And it's really going in and being able to understand the systems that are involved in various different clinics or hospitals or various different places where actually all of this data is being generated, whether it's a registry, patient organizations, and actually seeing what's there and going back to previous comments about if the data is not usable to begin with, who's going to actually be able to interpret it. So there was a huge investment in understanding what those tools could look like to be able to start to synthesize the data, standardize it, and make sure that not just genomic data, but clinical data could also be interpreted and added together. And so we've been doing that for quite some time, and we're actually expert now at being able to pull in the clinical data, clean it up, make sure it's in the right format, and also adding that to the genetic data. Now, the one thing I will say, and this again echoes what other people have said already, there's not a huge amount of the data out there to actually find. And while I appreciate the comment previously about wanting a more widespread approach to medicine and therapeutics as a whole, I completely agree with that. And I do think we need to do population sequencing in order to be able to get down to a personalized approach. But we're still not quite there yet. And there was a report that we did a couple of years ago that actually looked at the landscape of genomic programs globally. And there's about 200 of them globally, which is very exciting. And the estimate is that by 2025, there'll be about 70 million genomes available. And see, that is actually the type of number that we need to be able to start to make some of these insights. So IQVIA is working very closely with many of these global genomics programs to be able to understand what the need is and whether or not it's tools that we have, technologies, or partnerships that will actually accelerate that. Sometimes it's as simple as being able to understand whether or not it's the right consent form, as we heard previously, that's needed for that. You know, we're expert in doing decentralized trials, much more expert now than we were even 2 years ago. How do we start thinking about implementing genomics into some of those things as well? And we have very strong links with patient advocacy groups, and these are a huge, conduit of experience and both individuals who can help shape the design of trials, but more importantly, be an advocate for what this actually means on a genetic level. I think the one thing that's really surprised me is that my mother for many years could never understand what I was doing. Now all of a sudden she's an expert because she can say the word variant in PCR test, and she's not the only one. Everyone else can say this now as well. So the demystification of genomics, I think, is also really helping people who've been in this space for a long time. And I think that comes with both excitement but also caution. And until we start to view that genetic data is just another data point, there's always going to be that difference between genomics data and everything else. And I think many of our systems exist to be able to understand the linkage between both clinical and the genomic data. There's just, I think, a societal aspect of not necessarily being able to accept that. So while I'm really excited about a lot of the technologies that are coming down the pipeline, we spend a lot of our time being the plumber in the background, kind of linking a lot of these things together and being able to generate the data and being able to produce it for individuals to use is something that we've been pioneering now for many years and are still, I would say, quite far advanced compared to others. So delighted to be able to share our data with anyone who's interested in using it and to helping with the trials.
Thank you very much, Jo. David, you've kind of lived the journey of moving a company from early product development through the R&D to kind of initially penetrating the market and then market spread. So you've lived that journey and you've lived the challenges of getting it to a patient. Could you perhaps talk about Congenica's journey, but also it'd be great to understand what you see as the key limitations in terms of getting it out there?
You leave the room.
Always the investor with the problem.
That's right. Yeah. Mike's staring at me, so don't answer that question. I mean, that's a broad question. And in fact, I just actually want to build on something that Jo said that I thought was really important, and in fact contradict what I said to my answer in the previous question, which is it's all about data. And of course, it isn't all about data. It's all about identifying the information in the data. And Jo also mentioned the projection of 70 million genomes over what time frame?
It's supposed to be 2025. I think that'll probably slip a little bit because of this thing called COVID.
Yeah, but in fact, there are a number of new technologies from incredibly well-funded companies that are going to give our colleagues from Illumina a real challenge. And what the new technology that is actually moving into beta testing and there are functional prototypes, I think they're going to blow that 70 million genome number completely out of the water. And the projections of the sub $10 whole genome, it's going to suddenly put the pressure on the cost of sample prep and extracting DNA. So the world that we're sort of moving away from, which is, can we sequence every genome? Can we sequence the whole genome, or do we need to look at an exome or a panel or something? In my opinion, that's all going to change. And then if you think about what you do with that genomic data, and if you take the 30x step 3 to 3 billion bases of the genome, that's an enormous amount of data to sift through. And one of the things, going back to Adrian's question, that Congenica was conceived in 2013 to do, was to build a platform that could take all that data and identify the actionable information. So in the case of a rare disease patient, 80% of rare disease patients typically monogenic, so there's one or a relatively simple cause that's hidden somewhere in the genome. We built a system a number of years ago that can ingest that data, find that causal variant in a relatively short period of time, whereas it takes a very well-trained human days, weeks, months. We can, in some cases, do that in minutes. But we can also apply that to other disease states. The goal, and perhaps the value that Congenica is providing, is we actually see the way that genomics is going at the moment is sort of mimicking the typical centralized laboratory model. So if any of you run labs or work in labs, what you do is you get the sample and you send the sample, put it in a courier's bag and you send it to the lab. You get it sits there, it gets processed typically overnight or in the morning, you get it back. Huge amount of weight and waiting and waste in that process. I think the real promise of genomics is how do you make that more of a individual and patient centered approach where you can easily imagine that on an electronic patient record, whether that's in a clinical study or in a primary care setting, a patient presents to the physician and you can imagine how many pieces of information are contained with that patient's genome. If an abstract of that genome was attached to the electronic patient record and you had the analytical capability to be able to run that assessment while you're sitting in front of the patient, that suddenly creates a hugely informed decision, whether it's providing disease diagnosis, characterization, whether you're providing pharmacogenomic data. Potentially you can identify some inherited risk factors or acquired risk factors. So I think the opportunity for genomics and what we're trying to do at Congenica is to make the whole analysis that currently is contained within a person who's very heavily trained, has access to a whole range of reference data sets. And we're going to upend that. In fact, we have upended it by curating the data and making the data available so the whole process is automated and can be run on a phone, it can be run on a laptop, run on a desktop. So, you know, the journey for us has been partly getting the money to build that system, but it's also partly finding the partners and the physicians and the organizations who share that vision. And certainly 3 or 4 years ago, that was a difficult vision to sell. I think now, in a very short period of time, that whole concept of it's not about sequencing the genome and then hunting through it to find the information, it's how do you deliver that in a way that can be used on an everyday basis? Long answer to different questions.
Excellent. Matt?
Yeah, I'd also like to add to that. There's two aspects. There's finding the right target. So for that, you just need to sequence a lot of genomes, but not necessarily everybody's. Mm-hmm. And you've got enough to find the gene targets. But then there's also the question of when do you start to intervene with the patients who would benefit from a new therapy against that target? And there, in a lot of cases, if you wait until that patient presents with symptoms, you can slow progression or stop progression, but you can't usually reverse the damage that's already been done. So if we can get to a stage where we're routinely taking a whole genome of most people, it builds in the opportunity, and if you've got because that's a huge amount of data, but then if you can process that data and find those known targets before people present with symptoms, you've got a chance to actually block damage occurring in the first place. So there's those two aspects to consider.
Matt, can I just sort of go back to your earlier comments? You talked about the role of genetics and genomics in target validation. And we've built up an enormous body of data in the last 6-7 years that basically says if you develop a medicine against a target that has strong genetic linkage to the underlying disease, you're somewhere between 2-fold and 6-fold, or if you listen to AstraZeneca, 7-fold, more likely for that medicine to be approved. So it becomes a potential game changer. Do you take forward targets that don't have genetic evidence for association?
Yeah, we do. You know, really what we look for is validation of the target, and it's how strongly validated that target is. So if there's really strong evidence from, small molecule probes or siRNAs, well, siRNAs, it would be genomically identified target, but small molecule probes or something like that you can say, okay, if you can engage this target, you will get a positive outcome, and there's no genetic linkage. That wouldn't be a block for us. It's really the positive story of why do we believe in this target that matters. And genomic data is very strong evidence, so it's definitely a big plus. The other big advantage of having a genetic linkage is that opens the possibility of selecting patients for your trials. So again, you can start to exclude patients from those trials who aren't going to benefit from that intervention. And that has two benefits. One, it increases the power of your trial because you're more likely to see that delta, but also you're not exposing people to an agent that's got no chance of helping them. So it's a big plus to have a genetic linkage, but it's not a absolute no, we won't take it forward. Yeah.
Okay. Now, inevitably, data comes up again and again. So I'm just going to kind of turn back to our investors here. I remember being on a Genesis panel with Tom Hume a few years ago, and his view was if you're going to have data as part of your business model as a small company, make sure it's proprietary. Now, we have enormous public data sets. How do you kind of balance the value between applying proprietary analytics to public data sets versus having your own proprietary data as part of that aggregated data set?
I always recommend companies to have a dual strategy when it comes to that, because when you have something proprietary, you're effectively like building on a algorithm and then potentially using publicly available data sets to test the validity of your algorithm. And you probably have seen Tempest, right? They're sitting on a fair amount of data sets, and now they partner with some of the startups that I've invested in and ask them to build algorithms or test their algorithms on their data set and effectively co-market it together. as a diagnostics with a 50-50 sort of like profit sharing business model. So there are business models, new business models emerging from sharing proprietary data sets, allowing other people to validate their algorithms on top of that. One of my key concerns, though, which kind of touches back to your previous question, is around diversity in data sets. So a lot of genomic data sets that these algorithms are built on are built on Caucasian data sets, and that's something that is of concern because that's not the representation of the world as it stands today. So I think when it comes to data sets, I think there are interesting business models to be looked at, but I think there's the dark side that these data sets are excluding populations, or there's a missed opportunity there anyway.
So whilst it's clearly wrong from a social and ethical perspective to not have diverse data sets, if it's in the context of targets and drugs, where you're selecting patients based on a specific genetic marker or signature, does it matter?
It does matter, and you know what? And I'll say this because if you look at some of the companies that are coming out of Africa, which has the highest sort of like genetic diversity in terms of their population, there are some incredibly incredible insights to be drawn from those data sets. But you know, when you look at the companies that have been funded, say for example like 54Gene, they're effectively using Caucasian algorithms on an African data set. What needs to happen is new algorithms being built on a data set that is produced in the continent. So this is something that we're looking at as well in terms of, when it comes to understanding new biology, new kind of like targets as well. Like, I do think this is something that's important.
Thank you. Mike?
I wish I could say something controversial, but I totally agree with everything Anya
I was
said,
trying to be a little bit controversial.
Yeah, no, it was
Sorry,
good
that's
answer.
me.
Yeah. So I don't have much to add, and I David will comment. Congenica's strategy has been very much as you've described, which is relying on both the knowledge base as well as proprietary data. So you have to do both. And David's done a great job of making sure we do have partnerships in the Middle East and do have partnerships in Asia and other, not just relying on UK data. So it's a very good point. We're far away from solving it, so we need to keep on pushing that.
And if I was to push the other way, I'd say I don't think a continent wide. I think that's too wide, actually. One people are not. There are many, many, many diverse peoples within a continent in that sense. Okay, I'm going to learn from Mike, and I'm not going to continue my questions until I turn it over to you guys. Does anyone in the audience have something to ask the panel?
I was very interested to see a deal between Genentech and Microbiotica. And in this deal, Genentech are looking into IBD patients and are trying to profile the microbiome status of the patients to see whether there are any preferred patients that respond better. Are we going to see a lot of long-read metagenomic sequencing of the microbiome in clinical trials to look at the effect of the microbiome on patient outcomes?
I'll start because Microbiotica is one of our portfolio companies and we're believers in the microbiome. I think the way you've asked the question probably implies the answer, which is I think yes. The simple facts are, as human beings, we're actually more bacteria than human cells. And so you have to believe that the microbiome is going to play a really important part in our health. And there's lots of compelling data that shows how you respond to a drug, whether it's an IBD drug or a cancer drug, is massively dependent on the composition of your microbiome. And so the concept of adjusting your microbiome, or at least testing your microbiome and doing deep sequencing, will I think provide much better therapeutic results and patient selection. Maybe, I'm not sure.
Yeah, so I guess I'll chip in here as the world's largest CRO. I can assure you right now it's already happening.
Other questions from the audience? No.
So you mentioned the importance of the mining microbiome and also the fact that most of the, not microbiome, genomic sequence, and the fact that most of the monogenic diseases are less than 1% or 1% of the population. So how far we are in understanding, in mining the genomic to understand the most of our diseases which are of polygenic origin and understanding the targets there. I think there are some pioneering work in cardiovascular space. How about the other diseases and how much you see companies in this field?
I'm happy to answer
Yeah,
part
please.
of it. So with whole genome sequencing, we've seen how it's affected diagnosis of rare diseases in particular, which have been hard to detect at an early stage, and also less understood. So we've seen how whole genome sequencing has had a massive impact on patients with these types of conditions. But I don't know if anyone else wanted to take
the whole question.
I was going to say, I didn't quite hear the whole question. Could you?
So the interpretation of the polygenic diseases and the help, how it really helps us in therapeutics and development of therapeutics. Do you have any kind of view on how do we really progress there?
Well, I think one of the features of polygenic diseases is, in many cases, patients will fall into segments where they have similar profiles of causes, and my own personal feeling is that personalized medicine is probably going to be less about individuals and more about more and more segmentation of patient groups. So for different treatment paradigms, different genetic profiles might benefit more or less to certain interventions. And I think that's really probably one of the early places where you'll see consideration of polygenic causes coming into treatment.
I think there was part of the question on monogenic diseases, but we're at the very, very beginning. So that if you try and search through the literature and speak to people who know, and you say how many of the likely 3 million disease-causing variants have been catalogued, characterized, you can put them in a curated list. It's between 1% and 4% or 5%. So, we really are at the tip of the iceberg. And the probability of that remaining 95% actually being single nucleotide polymorphisms or even structural variants, it's going to be a chunk of it, but there are probably other factors, as Mike alluded to earlier, that are either in other parts of the genome or other types of modification. So I think even though, my earlier comment that we benefit from a massive genomics industry today, tens of billions in relatively simple analyses with simple analytes, sort of the journey into the more complex genomics for polygenic and monogenic diseases is really just beginning. But, there is a tremendous amount that can be done even with what we know today compared to, what we knew 5-10 years ago. But, I think it'd be a brave person who would claim to sort of see the end to understanding polygenic diseases in the next few years. I think it's a pretty long journey.
If there are no hands up, I'm going to ask a UK PLC question. So, we talk about it a lot. We talk about the fact that we have this ecosystem, right? We have a fantastic National Health Service. We have organisations like Genomics England and very large data sets, Biobank, HDR UK, really healthy investment at the moment, really healthy investment, cohorts of SMEs, real capability in technology. So there's this big functional genomics opportunity sitting there, how we start to actually move from data to utility of data and targets and all the other things that will come out of it. But equally, we have this position where like the whole is less than the sum of the parts. what do we need to do? What do we need to change to really kind of optimise the functional genomics opportunities in the UK? And I'll open up to anyone on the panel to start.
Adrian, why don't you share your thoughts on that?
So my thoughts are pretty broad. If you go from the base, I think we need to make... So when I think functional genomics, I do think about tools and capabilities. I think about single cell. I think about cheap sequencing. I think about interpretation tools and algorithms. But I also think about things like models at scale. You know, thousands of organoids across multiple diseases of complexity. Because it's about getting representative models at scale. It's about perturbing them at scale. It's then about feeding that data and aggregating it with other global data sets and then interpreting it to pull out whatever your particular area of interest is. I think academia fails in some respects. It doesn't fail in what it generates, it fails in how it disseminates it and how it interacts with industry. Yeah. I think some of the fantastic data sets from organisations like Genomics England, we haven't actually thought, at two levels actually, we haven't thought about how to maximise the distribution and use of those, and we really haven't thought about how to bring the SME community into helping to create value from those. You know, it's this kind of make or buy type of opportunity. NHS adoption of technologies has always been difficult, and it will always have complexities that I can't speak to, but it's not efficient. So actually I see multiple barriers across the entire chain. You know, as a starting point, we need to make our data sets freely available and easy for people to get their own analytical tools into those domains and take their own findings out of it. So that'll be my kind of starter for 10.
Yeah, and the reason I ask Adrian that is, you know, we're both members of the BIA, and in fact I have the pleasure of being on the BIA board, and I'm the BIA representative on the National Genomics Board. And you know, I'll say something that is both encouraging and potentially frustrating, but certainly the commitment from some of the senior leaders within public and private genomics within the UK are completely committed to implementing the vision that's outlined within Genome UK that many of you have seen. I think the challenge is all those bits in between. You know, the amount of money that's getting lined up to be invested in the public programs, so Genomics England, Our Future Health, UK Biobank, you know, they're just three of many programs that are without any doubt the envy of countries around the world. But I don't have the answer, but the question is, how does that trickle down, and how does that stimulate the entire ecosystem? So we go from 250 companies, I think it is, or somewhere around that, to 500 to 1,000. So there's a lot of good things happening, but the question I think everybody asks is it happening fast enough? And is the benefit impacting a broader group of stakeholders?
Yeah. I'll just chip in and say, I think one of the things, and this goes back to many of the points that were raised earlier, is it's great to have this ecosystem in the UK, but we need to share. Right, it's fantastic that we're actually leaps and bounds ahead. I was just saying to David, I was in Turkey last week. The Turkish Genome Project, it was announced, I think, before or perhaps around the same time that we announced our initiative in the UK, and they've sequenced less than 1,000 individuals. So what's the difference? Is it money? Is it political will? What is it? What can we be doing to share? Again, going back to very basic principles, is it a consent form? There's no cookbook that says how to do this, but we actually have done it. So I think the UK is responsible in some ways to be able to share that information, and also, as Adrian said, share the data. So how can we be a bit wiser about that? The other thing I'll also say is that while genomics is clearly the best omic out there, there's other things coming down the pipeline that are going to be complementary, and I don't think we should dismiss some of those capabilities and being able to line that up. So where can we get to with transcriptomics, for example, and various other omics? And I think we need to be cognizant of that, and actually maybe it's not doing more, is it taking subsets and supplementing it in a slightly different way to gather insights, and again, to then share that globally.
And I think that the business model is probably going to need to evolve as the technology, because at the moment we're still in the process of developing the infrastructure to actually create the data in the first place, and then to understand it, because there's so much data from genomics and all the other omics. And then the question is going to be, as that becomes more and more routine, the question is going to be, are big pharma companies going to take that in-house and set up their own units? And will it become another tool, or will it always stay a specialist discipline, which is, we have standalone companies providing that service into the companies, and I think we just have to see how the field evolves.
Matt, do you think there's a different boundary for what's pre-competitive versus what's competitive?
Yeah, that's an interesting question. I have a feeling that probably the raw data, just the sequences and the distribution spreads, it's going to be hard to keep that out of the public domain. I think that's going to end up in the public domain. And I think the competitive advantage is going to be how innovatively you can use that, how you can turn that information into insight and then into progress. I think that's where the real competitive differentiation is going to lie.
I think we've run just slightly over. Okay, thank you very much, and please thank the panel here.